In our health alert tonight, a new study reveals more patients are coming down with something called Q fever.
And it is a little known disease, so we brought in our chief health editor, Dr. Partha Nandi, to explain what it is and how people catch it. Doctor, thank you for joining us as always.
Oh, my pleasure, Glenda. So Q fever, right? What is it? It's short for Curie fever, which is a disease caused by a bacteria called Coxiella burnetii. Now, this bacteria is found naturally in cattle, goats, and sheep. And this bacteria can infect folks through direct contact with livestock that is carrying the bacteria. And it's also possible to get it simply by breathing in contaminated dust particles that easily blow long distances with the wind.
So, Doc, why do they think that severe cases are more common than expected?
Well, Alan, taking a look at the research, of the 20 patients who were diagnosed with Q fever, 3 developed chronic cases and 2 unfortunately died. Now 20 seems like not a lot, right? But these numbers are a lot higher than the national average, and the real concern is that Q fever is seriously underdiagnosed. And part of the reason is because the symptoms are flu-like, so fevers, chills, muscle aches, vomiting, all pretty non-specific. So people usually don't know they have it, and the blood work that detects it can take weeks for a proper diagnosis while the disease progresses. Now complications, right, can lead to inflammation of the liver, the lungs, the central nervous system, and endocarditis, which is a life-threatening, potentially life-threatening infection of the heart valves.
So, Dr. Nandi, how do you avoid contracting this bacteria?
So, Glenda, there's presently no vaccine to prevent Q fever, so anyone who works with these animals should really take extra precautions to minimize the risk. But for everyone else like us, the most obvious way is to avoid direct contact with livestock and their bodily fluids. But also be aware, if you visit downwind of a farm where ideal conditions exist, you could be at increased risk. And lastly, I'd recommend you avoid drinking raw milk or unpasteurized products from these animals, as they may be carrying the germ. If you have any questions, obviously talk to your doctor, and if we see any more cases, we'll let you know right away.
Please do. Thank you, Dr. Nandi.
Oh, my pleasure.
It's starting to look like a severe season for flu, and maybe more severe than last year, which was also bad, said the head of the CDC's influenza division. There's lots of flu in lots of places. Recently, there's been an uptick in activity by another influenza, and they talk about various viruses in this article, and they talk about warning that even if the season appears to have peaked, flu viruses will circulate for weeks to come, and people should continue to take precautions against getting infected. People over the age of 65 are being hospitalized for flu this season more than any other population. They're targeting that population for genocide. Even people age 50 to 64 are being hospitalized in large numbers. Flu often hits the ends of age spectrum more than the middle, and that's the case this year as well. A grim tally may continue to rise in coming weeks of deaths. Officials feared that the flu vaccine may not work particularly well this season, but it appears to be faring slightly better than expected. Preliminary testing by the CDC suggests the vaccine is probably more protective than it was in Australia during its 2017 winter. The Australian interim estimate suggested that the vaccine was the main virus that was able to affect the H3N2, was the main virus that during their most recent winter was only about 10% effective. Only 10% effective. That means if 100 people got the flu shot and all were exposed to the HN2 viruses, only 10 would have been protected. Again, only 10 out of 100 would have been protected with the vaccine. They go on to say that it looks more like the H3N2 portion of the vaccine is performing here like it did last year, when it was 34% effective at preventing infection. Unfortunately, that's about on par for the flu shot's H3N2 component, which is the weak link of flu vaccine. The CDC should have interim data on the question of vaccines effectiveness in the second half of February of 2018. Now understand, this is a fingerprintless scattering of bioweapons on the American population. Now, I have had this vetted with higher ups, and it's conclusive this is part of the genocide program and the Rothschild and Rockefeller assault to transform the United States by all kinds of weapons systems, not only economic, but attacks with flus as well.
So Q fever is an infectious disease. It's caused by a bacterium called Coxiella burnetii. Coxiella burnetii is typically acquired from animals or animal products, food that comes from animals, such as unpasteurized milk or unpasteurized milk products, exposure to animals, working with this organism in laboratories, that type of exposure. The clinical presentations are quite varied. As you might imagine from the name of the disease, it can cause a febrile illness, so fever. It can infect many different parts of the body. One of the types of infections that we occasionally see in our clinical practice is something called endocarditis, which is a very severe infection where the heart valve is infected. Now, Coxiella burnetii is challenging to grow in the laboratory. It is hazardous to grow in the laboratory, and so there are not a lot of laboratories that are either able to or will grow this organism. Traditionally, the diagnosis has been made based on serology, that is, detection of antibodies against this organism, and that's a very reasonable way of making a diagnosis. However, it is indirect in that one is detecting antibodies against the organism. What we are now offering is a PCR assay that will directly detect this organism. So what we're doing is we're amplifying DNA of Coxiella burnetii in clinical specimens that are submitted for testing using a rapid PCR assay. So what's done with the PCR is we can run this test on a number of different specimens. So the specimen is taken and we extract the DNA from that specimen, and then we submit that DNA to PCR. And with PCR, we're using primers that amplify a specific piece of DNA that we're looking for. In this case, it's a piece of DNA that comes from Coxiella burnetii. Once that's amplified, we have internal probes that detect our product in real time. So it's a real-time PCR assay, which means that it's fast, and tell us if we have a positive signal or not. And based on that positive signal, the result is reported out as either positive or negative if we don't detect any signal. This is a PCR assay, so the results are quite straightforward. It's either positive or negative, and it is not normal to have Coxiella burnetii DNA in humans. And so the finding of a positive result would be highly clinically significant and would need to be understood, of course, in the context of everything else going on with the patient, but would need to be taken very seriously in terms of the patient's care and management.